go with today's PhD works and all. Not only that, even the projects. Just simply analyzing the drug formulation is not enough. We need to go for clinical side, and we have to understand the bioavailability, pharmacokinetics, all these things. And that's why, as per PCI syllabus, we have all these topics like pharmacokinetics, toxicokinetics. So we thought of including this topic by Sutik's HOD, Barney is Sogalika. And I welcome ma'am. Actually, it's very embarrassing to say ma'am.
Don't call it also.
yeah. She's my colleague, that's why. Yeah. Thank you, Barani.
Thank you, Vijaya, for giving this opportunity.
Yeah, I request Pratyusha ma'am to give a brief biodata of our speaker.
Okay, ma'am. A cheerful morning to dear staff, students, and speaker, ma'am. Myself, Maunika Pratyusha S., Assistant Professor in the Department of Pharmaceutical Analysis. I would like to give a brief introduction about our today's dynamic speaker, Dr. Barney S. Sogali, ma'am. Ma'am has completed her graduation in Sri Padmavati Mahila Vishwavidyalayam, Tirupati. Ma'am got her PhD and PG from AU University, Visakhapatnam. Ma'am also completed Executive MBA from National Institute of Business Management, Chennai. Ma'am has an outstanding teaching and research experience for more than 14 years and also guided around 17 MPharm projects. Ma'am has around 34 research publications in both national as well as international journals. Ma'am acted as an active member for MAC Power Independent Ethics Committee between 2008 to 2009 and reviewed and also approved 5 protocols. Ma'am is a reviewer and assistant editor for Journal of Pharmaceutical Research and editorial member for Indo-American Pharmaceutical Journal Sciences. Ma'am worked as chief editor of APTI Women's Forum newsletter from 2016 to 2019. Ma'am has a great knowledge on GCP, good clinical practices, and involved in giving training on spray dryer in RGUHS workshop conducted by Krupanidhi College of Pharmacy. With this brief introduction, I would take the privilege to introduce Dr. Barani S. Sogali, ma'am. Ma'am, I request you to start today's session and take us through the journey of enlightenment and knowledge. Thank you.
Thank you. Thank you for your warm welcome and inviting me and giving me this opportunity.
Thank you, ma'am.
To share my knowledge on pharmacokinetics. Okay, I'll start. Can I start sharing, Dr. Vijaya?
Sure.
Shall I start?
Sure.
Participants are only means they have.
Yeah, that's what. Anyway, they are going to join. Like we will wait for 2 minutes, 2 more minutes because it's 10:28.
Sure.
Yeah. Thank you.
Okay, shall we start? Okay. Actually, Dr. Vijaya already mentioned why this pharmacokinetics topic in bioanalysis workshop, right? So that there is an answer because all these are interrelated topics actually. So in bioanalysis, you will be analyzing the samples from various physiological fluids, biological fluids, serum, plasma samples and all. And you will be doing assay and determine the quantitative discover quantitative method, how to estimate that sample. So what samples you are analyzing? You will be analyzing the all the fluid samples, plasma samples and all, from where this data, where these samples are coming from. Because once the drug is administered into animals or humans, the samples will be connected and will be sent to the bioanalytical department. So you will be analyzing and giving the concentration and what is the lower limit of quantification and upper limit and all the drug details, how much is the concentration, everything you are going to give us the data. So as a pharmacokinetic member or as a pharmacokinetic person, what we had to do is we had to analyze and estimate the concentration, Cmax, various pharmacokinetic parameters. So this is all interlink is there between pharmacokinetics and clinical studies, bioanalysis, and pharmacokinetics. Once the data you give to pharmacokinetic department, so they will be analyzing various parameters and assessing the data. So where this data is going, what we are going to analyze, and what is the, how this data is useful. Okay, we'll get various pharmacokinetic parameters once all the data is we gather from the department and analysis department. So where this data is going and what we are going to do with this data, all that things we are going to cover in this today's session. And I request all the participants to be active in the session. Whatever you have doubts, you can ask in the chat. And if I ask some questions, please, you can share in the chat. No need to answer, no need to raise your hands, nothing. Just you can chat in the form of whatever the answers you are going to say. I want everybody to actively involve in the presentation. And please clarify your doubts if you have any during the session or after the session also. I'll be happy to clarify your doubts. So today's topic is application of pharmacokinetics in drug development. So I want to just ask, what is this pharmacokinetics? Maybe you would have in the pharm analysis PG students, I think you would have forgotten what is pharmacokinetics because you would have studied in B.Pharm final year, right? So what is this pharmacokinetics? It is the time course of ADME process. ADME process means absorption, distribution, metabolism, and elimination. So what this disposition, where the drug means, we can say in the other words, like what the body is doing to the drug is nothing but pharmacokinetics, right? So here, passage of drug from the site of administration to the systemic circulation. You know, I think all of you may be knowing this. Once the drug enters into the blood, that process is called absorption. And where this drug, once it absorbs, where it goes into the different body parts, that is, it is getting distributed. So, and it distributes, during distribution, it binds to various plasma proteins also. So protein binding followed by target specific and non-specific tissue uptake. So that is distribution. So once the distribution happens, it shows its own action and everything. Then once the action is shown, the drug is getting metabolized. That is phase 1 metabolism is there, phase 2 metabolism is there, and then the drug will be eliminated. That is removal of intact drug. So this is all together about pharmacokinetics. It is the study of how the drug, what the body is doing to the drug. So once the drug is administered, it goes into the systemic circulation and gets distributed into different body parts. Then it gets metabolized and gets eliminated, right? So any doubts till this? Okay, next. We'll go to the next slide. So this is the pharmacokinetic profile. I think we study in biopharmaceutics 4th year B.Pharm. So this profile is nothing but pharmacokinetic profile we say. So here, once the drug is given and it reaches into the systemic circulation, so it reaches to a concentration called Cmax. That is the maximum concentration of the drug in the plasma. So once it is reaching the Cmax, slowly it gets distribution and elimination happens. And this phase is called elimination phase. And this is called absorption phase. And this is completely the area under the curve represents the concentration of the drug in the plasma during the particular given period of time. And this is the maximum therapeutic concentration, and this is minimum effective concentration. So this much concentration should be there in the plasma to show its action on the body. So, and this is maximum therapeutic concentration. Once it is this MTC, definitely it will start some toxic interactions, and it leads to toxic reactions in the body. So between these MEC and minimum effective concentration and maximum therapeutic concentration, there is a therapeutic range. And during this Cmax till the distribution happens, that is duration of drug action. And the time to reach the maximum concentration in plasma is Cmax. Okay, so this is a complete, I'm giving just basics so that when we go to into the depth, you should be able to understand what are these parameters, what I am discussing. So next one is pharmacokinetic parameters. What parameters we are going to assess once the data is we get from the analysis department. So here pharmacokinetic parameters are assessed by monitoring variations in the concentrations of the drug and its metabolites in the physiological fluids that are easy to access. Especially we are going to analyze plasma samples, one, how the drug is getting distributed and how much time it is reaching to the maximum concentration. All these data will be analyzed, and then how it is getting excreted, we are going to analyze using urine samples. Means how the drug is getting excreted, how much is getting excreted, everything will be getting to know from the urine data. And plasma concentrations are usually checked, and if required, additional biopsies also will be tested, and sometimes some human studies also. So what these parameters give an idea? It gives overall indication of the behavior of a drug in the body. So for pharmacokinetic parameters, all this data, once we are going to assess after the concentration data we get from the analysis department, is behavior of drug in the body. So what are the parameters we are going to assess during, generally after IV administration? AUC, plasma clearance, volume of distribution, and elimination half-life. And if it is given orally, we are going to discuss, take out the measurement of lag time. What is this lag time? The amount of immediately when we give the drug to the body, and it will not absorb immediately if we give extravascular or oral, right? So when we give through intravascular, definitely the drug reaches immediately into the blood and no absorption process will be there. But when we give orally, there will be a lag time will be there till before the drug reaches to a measurable concentration in the plasma. So then Cmax we are going to assess, Tmax, AUC, and elimination half-life. These parameters we have to assess. And the parameters obtained from urine concentrations, whatever the route of administration, whether it is oral or whether it is IV or whether it is IV infusion, whatever the administration in material of it, we are going to assess amount excreted in the urine and renal clearance when plasma concentrations are known. So these parameters we are going to assess as a pharmacokinetic data minimum from the various samples. So mainly we are going to estimate other 3 parameters which are very, very important parameters to outcome. What is to describe the outcome of ADME factors is nothing but 3 important one: volume of distribution, bioavailability, and clearance. Volume of distribution is amount of drug in the body divided by plasma concentration. What is the amount of drug which is getting distributed in the entire plasma and the different body organs is nothing but volume of distribution. So how we are going to calculate this volume of distribution is the drug divided, the amount of drug concentration divided by plasma concentration. And bioavailability is nothing but completely about the how much concentration of drug is reaching the systemic circulation. Usually IV means 100% drug will be reaching, and other routes of administration, it depends and it shows variation. So clearance, it is the how much drug is getting eliminated, what is the rate of drug to get eliminated. So that is calculated by multiplying the elimination rate constant into volume of distribution, and generally it is measured in ml per minute. These are parameters we are going to assess. Okay, bioavailability, Cmax, Tmax, lag time, and onset Tmax time. So all these data we are going to calculate using nowadays many pharmacokinetic softwares are available. Once the data we get from the analysis department, we are going to put into the software and analyze all these data. And this data, where it will be useful? That is nothing but today's topic, that is application of pharmacokinetics. So application of pharmacokinetics in drug development, right? So how the drug is getting developed. So I think I am going to just focus on a brief graphical representation here. So here, during the drug is discovered, during drug discovery in R&D department, some almost 5,000 to 10,000 compounds will be tested. In that, only 250 compounds, or nearly 200 to 250, or even sometimes less than 100 compounds will be eligible for the next, that is preclinical studies. Preclinical studies means if the drug is showing action on that particular indication, what we are testing for, so then it will go to the preclinical studies. Preclinical studies means animal studies, or we can do testing in the biopsy or some cell lines and different fluids, biological fluids. We can collect saliva samples, we can get plasma samples, and drug we will see, and how it is getting ADME pathway we are going to assess. That is preclinical study, that is animal studies, and around 200 to 250 compounds only will be eligible for this preclinical study. So once this preclinical study, we are going to assess out of 250, nearly 5 to 6 compounds will be eligible for, and they will show completely action for what we are looking for. So these 5 compounds will be selected for the further trials, that is called clinical trials. So once the animal studies are proven some positive response, and it is showing some positivity among the existing or marketed preparations, if it is showing some positive reaction towards the particular disease, then definitely it will be taking for this clinical trials. So clinical trials means, what is this clinical trial? We will be testing on humans. So if it is tested on animals, that is preclinical study, and clinical studies or clinical trials means we will be testing on humans. So here phase 1 studies, phase 2 and phase 3 studies. There are 3 important phase studies are there in the clinical trials, phase 1, 2 and 3. And I will go much in detail in the next coming slides. So once the clinical trials show positive response, and it shows some definitely value to the indication for what we are testing for, definitely it will go to the FDA approvals and regulatory approvals and new drug applications. So once the FDA gives approval, that is comes under new drug application, and here till the before clinical trials, IND means investigational new drug, the clinical trials are conducted under this IND application, that is investigation new drug. Once the drug is proven very successful in clinical trials, and then it will go to the NDA submission, that is new drug application, and the product will be manufactured to a higher scale up. They will be scaling up and increase the production. So once the product is released into the market, after the FDA approval, post-marketing surveillance is there. So this also, it covers under phase 4 study, which we say, that is post-marketing surveillance, and it is indefinite in a number of patients. It is a continuous study. Once the drug is released into the market, we will be testing again on humans some occasionally, and we will see any adverse events are happening after administration of this NDA to the patients, and it will be done in a large number of population. So we'll discuss one by one what is this clinical trials, and phase 1, phase 2, phase 3, and where this PK data is fitting in, and how it is useful. So drug development builds on pharmacokinetics only. So pharmacokinetics means even bioanalytical department also is equally important in this. So once the drug is developed, so here discovery and development to a preclinical research, clinical development, which I told that there are different trials, phase 1, phase 2 and phase 3, to FDA review and post-market monitoring. All these steps, or all one by one stepwise, if you see, all these studies will be collecting the samples from the patients, from the animals, from the patients, and testing all the PK parameters for safety and efficacy. For what purpose we are testing this pharmacokinetics on human studies or in human population or in animals? It is just to assess the safety and efficacy of the drug. Once the safety and efficacy is successful, that the drug is very efficacious and very safe at lower doses without any much toxic or side effects, then definitely it will go to the next step, that is marketing, phase 4 studies. Okay, so this is overall PK pharmacokinetics is applicable throughout the drug development till it reaches into the starting from the preclinical to the post-marketing till it reaches the phase 4 studies. Entirely the pharmacokinetics play a very vital role. So if you see here drug development stages, preclinical phase 1 trial. So in the preclinical, we identify a target and drug and we do the in vitro testing and then we will test on animals. So phase 1 initial safety evaluation is done, and phase 2 efficacy evaluation is done. Phase 3 test and efficacy both will be tested, and phase 4 ongoing. It keep on we had to keep on testing once the drug releases into the market. Please don't think that, okay, the drug is released into the market, so no need to test anything again further on the data. No, we had to continuously monitor the data. We had to collect some samples from the patients, and we need to see the adverse events, if any adverse events are happening, and we need to submit to the regulatory for the continuous update. So here, if you see phase 1, there are different phases are there in the clinical trials. Phase 1, phase 2, phase 3, and phase 4. So this diagrammatic representation you can only you can assess. If you see, phase 1 is only on a small number of population, that too on healthy volunteers. I'll go much in detail in the next slide. So phase 1 is human pharmacology. Phase 2 is therapeutic exploratory. Phase 3 is therapeutic confirmatory. Phase 4 is post-marketing studies. So here, if you see, phase 1 is on lower population, it will be tested. Phase 2, it will be tested on a group of patients, a little bit higher the number than the phase 1. Then phase 3 will be tested on large number of patients. And phase 4, it will be like population pharmacokinetics. The drug will be marketed throughout the country or region, and we had to monitor throughout the studies. Till now, any doubts or anything, you can just please chat so that I can clarify. Okay. So here, phase 1 studies. What is this phase 1? Once the drug is proven that it is very successful in the particular, for whatever the indication we are trying for, like cancer, or it is for some elderly people or anybody, for whatever the indication. So phase 1 trials will be tested first. So here pharmacokinetics again play an important role, because it is a transition from animal studies to human studies. Okay, and single most important event it will be tested. So here the next slide I'll be telling this. It starts at lower doses. First we will give whatever based on preclinical data. What is the minimum dose can be given without any toxic reactions to the patient, that will be assessed. And that much low dose will be given to the patients. And I want to correct here, the phase 1 trials will be tested on healthy volunteers, not on the patients, just to measure how the drug is getting absorbed, and how the drug is, how much time it is taking to reach maximum concentration, and whether the drug is safe or not. Only this will be assessed in phase 1. So it starts at lower doses and escalating as a safe to therapeutic range. If it is proven in the initial study that the drug is very successful and it is very safe and it is not producing any toxic reactions, then we had to increase the dose, that is super therapeutic range, means above the therapeutic range also the volunteers will be administered the higher concentrations, and we will find the best dose suitable for that particular disease. And the phase 1 trials will be tested on small number of subjects, that is around 20 to 80. And there are single ascending dose studies and multiple ascending dose studies will be done. So this I'll be discussing in the last slides also, that the SAD and MAD studies. Okay, so phase 1 will be starting at the lower dose and a small number of volunteers, and generally 20 to 80 subjects. And There are SAD studies, that is single ascending dose and multiple ascending dose. And overall, the phase 1 trials gives the conclusion that whether the drug is safe at a given particular dose and what will be the best dose to produce its action completely to treat that particular disease will be assessed. Okay, for this, where the pharmacokinetic parameters, why she is discussing all these pharmacokinetic parameters and all, but she is telling phase 1 trials and phase 2 trials, you'll be thinking that. So one here also, once the data samples will be collected from humans, like plasma or urine samples or whatever the samples we collect from the patient, we had to analyze it through bioanalysis department. And then once the data is collected, we are going to assess all the Cmax, Tmax, everything. And based on that data only, we are going to show that the drug is safe or not. So data, pharmacokinetic data plays a very vital role. Next, phase 2. So unlike phase 1 we are testing on volunteers, and here phase 2 first trials in patients in which the disease to be treated. For example, if we are, our medicine is oncology or means against cancer, so we had to test on patients with cancer in phase 2 trials. Phase 1 means only on human volunteers. So here to evaluate drug in particular indication or disease, so common short-term side effects and risks with IND will be seen in this phase 2. And especially dosage regimen for phase 3. So during phase 2 studies, we are going to analyze what is the dose, next dose to be given, so that the drug will be maintaining the particular concentration in the plasma. And here the patients will be in little larger number compared to phase 1, that is 50 to 300 patients, participants will be testing. And what is the outcome of this phase 2 trials? Once all the PK data is given, then we are going to assess the safety and efficacy of investigational new drug. So it may take weeks to, it may take 2 months. Then during the phase 3 trials, once the data comes from phase 2 and phase 1 and preclinical studies and all the things, we are going to just confirmatory trial it is. We are going to confirm whether the drug is particularly like cancer or any other disease. So whether it is showing complete reaction, what is the best dose, which was decided already in the phase 2, whether it is just to confirm this, phase 3 trials will be conducted. And it is for preventive or therapeutic effects, and in large number of patients, nearly 1,000 to 5,000 patients, we are going to test the investigational new drug in the patients. Once once it is proven that this is very confirmative and showing very efficient and very safe, then it will be then we are going to file for NDA. And then NDA, once we get the approvals from FDA and all, then we are going to release into the market. And once the marketing, after marketing, phase 4 trials we have to perform, that is post-marketing surveillance. Post-marketing surveillance means once the drug is released into the market, we are going to assess in all the entire hospitals, wherever the drug is getting prescribed. So we are going to monitor this, how the patients, whether any adverse events are happening or very serious events. All this data will be collected from the patients and continuous analysis will be done. So effective early drug development, phase 1, phase 2, actually phase 2 trials are again covered under phase 2E and phase 2B, and phase 3 and NDA filing. So here, where this PK data or PD data is applicable. So here, if you see single dose safety, single dose, only one dose we give to the patient, and we are going to assess what is the safest dose to be given to the patient. And tolerance, what up to what tolerable dose the patient's body can able to withstand, that is tolerance. And multiple dose safety, all these we are going to assess. And in the not only phase 1 is very, very critical trials for the new drug, that is investigational new drug. So because here we are going to assess multiple parameters, although it is on a small number of volunteers, we are going to assess what is the availability within the patient, what is the difference with within the and between the patient, between the volunteers, between the subjects, how it is showing variation, and what is IV dose to be given, what is the food effect, drug-drug interactions, age and gender, and renal and hepatic disease, multiple dose pharmacokinetic data in patients. After giving multiple doses, how this PK variation is happening. All this we are going to assess in phase 1. This I'll be covering in future coming slides also. So comes and we are going all the concentration of drug in the plasma or any given fluid we will be getting from the department analysis department. So we are going to fit into the suitable models to identify how the drug is getting distributed or metabolized. For this, we are going to use some analytical techniques in pharmacokinetics also. That is, if you remember, you would have studied in your final year, that is again compartmental modeling and non-compartmental kinetics. So non-compartmental analysis means estimates the exposures and elimination properties of the drug, especially how the drug is getting eliminated. If in this model, we are going to think that the drug is how the drug is distributing and coming out from the body, eliminating from the body. Okay, so compartmental analysis means we are going to think the entire body is divided into different compartments, and we are going to fit some mathematical expression or model which describes the pharmacokinetics of a particular drug. So predictive of PK at different dose levels or administration conditions. So compartmental modeling is little complex models which we should use in elucidation, which make up the pharmacokinetics and this entity, such as rate and capacity of different metabolism pathways involved in drug elimination. Especially almost 95% of this phase trials or like preclinical studies and all, they use non-compartmental analysis. However, if it is done in large population or like a single patient or number of patients are small, then we are going to adopt this non-compartmental analysis technique to estimate the data. And compartmental analysis, it is a little complex model. So we are going to use for when the population is large and we are going to test on large number of patients. So if you remember, this is the drug, we in one compartmental pharmacokinetic model, a graphical representation given in this slide, where we are going to think that the drug is getting into the single compartment, central compartment, and eliminated central compartment. If you see here, that is after IV injection dose into the single central compartment of volume V, and with the first order elimination constant, that is K elimination rate constant. Cp is the concentration in the compartment which decreases over time t. So because the drug concentration decreases over time because it is getting eliminated. So this we are going to assess in the one compartmental PK model. So population pharmacokinetics, what is this population pharmacokinetics? It is a systematic analysis of combined data from the specific studies or from the entire drug development program. So once the drug is proven in all the 3 trials and it is proven successful, then we are going to test on large number of population, whether the drug is effective and what is the population variability. PK variability studies also will be conducted. So how much is the variation happening from patient to patient? So that and all will be tested under population pharmacokinetics. If you see, there are large number of data, PK data is available here. Drug concentration versus time post-dose. And here we are going to see the solid line, which with 95% confidence interval with the observed data. This is all the scattered data, but we are going to see how much data is matching in a large group of patients. And we are going to assess this profiles. Based on this, we are going to assess how the drug is showing variation from patient to patient. So non-compartmental model parameters, we are going to assess Cmax, the time to reach maximum concentration, the time to reach maximum concentration, the area under the curve which represents the bioavailability, and what is the elimination rate constant, and what is the half-life, what is volume of distribution and clearance, that AUC infinite till infinite time. So all this we are going to assess apparent volume of distribution after extravascular dose calculated by this CL by, that is clearance by fraction of drug eliminated. So here, early clinical studies with primary PK endpoints. What is this early clinical studies? So this also comes under clinical studies only, but early only. That is nothing but phase 1 studies if you see. So phase 1 studies, as I told you, it is a very, very critical trial stage where we are going to assess the safety of the particular given drug under different conditions. Okay, so where the primary endpoints are all are PK related. So application of pharmacokinetics is everywhere in the entire drug development, starting from preclinical to the phase 1, phase 2, phase 3, and phase 4 also. All the data will be assessing. Overall, this PK data is used to evaluate the safety and efficacy of the given approved drug and also investigational new drug. Okay, so phase 1 studies will be described as studies where the safety measurements are the primary endpoint and where the primary endpoints are PK related, pharmacokinetic related only. And these studies are determine the effect of other drugs. What are the concomitant drugs which are given to the patient, and what particular disease, if we are going for a diabetic patient, and what other diseases he is suffering, and whether our drug, investigational drug, is effective in that conditions with other diseases also, in presence of other disease, how it is showing the action, how it is showing ADME distribution pathway, how it is changing, all this will be analyzed. And the patient qualities on the pharmacokinetics of a drug in relatively small number of subjects, reducing the risk to the patients. Why we had to do all this in phase 1, only in small number of volunteers? Because our main aim is to, so that if the drug is not proven that it is not effective or not safe at the given particular lower doses also, then there is no point in wasting our time in phase 2 and phase 3 studies. And we are exposing large number of patients in this phase 2 and phase 3 test studies, and we are giving more risk to benefit ratio will be more. So more risk only. So that's why, to avoid all these risk factors and to avoid the risk to the patients in phase 2 and phase 3 studies, and to avoid the exposure of drug to the further level, next to the next level, we are going to assess all this in early clinical studies. That is like phase 1, sometimes they call as 0 studies, phase 0 also. So especially just to evaluate the safety. So first in human studies, the main purpose of this first in human study means this is a transition. What I told is from animal studies directly we are going to test on humans. So that's why it is called first in human study. And it is to test that it is safe, meaning the subjects are monitored for signs of toxicity, whether any toxicity. Actually, all these toxicity studies we are going to finish in preclinical studies itself in animals. So once the drug is proven that it is not toxic at a MTC, maximum therapeutic concentration also, then only it will be taken to the clinical trials, that is early clinical studies. So though we are taking the drug after the toxicokinetic studies approval from preclinical, we had to test again in patients. In humans, we don't know how the drug is getting absorbed, distribution, and ADME pathway. So that's why we are going to test on patients for toxicity, a test on human volunteers or subjects, they say. So for the signs of toxicity, especially those indicating risk of mortality or morbidity. So all this we are going to assess in first in human studies. Then tolerability, that is the ability of patient. Sometimes this tolerability varies from patient to patient. Some patients can tolerate the maximum dose. Some patients are not tolerable even at the dose selected for them to be given for the appropriate action against the particular disease. So tolerability is the ability of the patient to use the drug for intended indication without unacceptable, non-life-threatening adverse events that would require discontinuation of the treatment. If the patient is not tolerable, then we cannot give the drug. We have to discontinue them from the treatment. So risk to benefit ratios are considered when determining the required tolerability and risk of toxicity. So a drug for life-saving, but this is not applicable for sometimes for life-saving drugs such as cancer would be considered for approval, even if it carries more risk for a drug as for a self-limiting or non-life-threatening disease such as common cold. Common cold means why to test the tolerable dose when the patient is not able to withstand the dose. No need to continue for phase 2 and phase 3. But the same, if it is a cancer effective and the patient may not able to tolerate the dose, but still we will go for the further studies so that the patient benefit will be there compared to the risk, because if the cancer is getting treated, though the patient is not tolerable, we can just reduce a little bit lower dose and we can just still use for the continue the further studies for phase 2 and phase 3. That is first in human studies. And as I told you in earlier only, in phase 1, all these only will come under. So phase 1 means early clinical studies, first in human studies, single ascending SAD, MAD studies, all these comes under phase 1 only. So FIA study is the single ascending dose study where small number of subjects are dosed carefully with either a drug or a placebo, and the safety is monitored. So we will give a small number of very low dose and we will see whether the drug is safe or not. And once it is proven it is safe at the given dose, then higher doses we have to administer. So because we have to see the tolerability also, right? So typically 2 to 3 times higher than the starting dose will be given. It is administered to a new group of subjects, which is then considered a higher at dose is given. So if the patient is able to tolerate the 3 times the safer dose also, then it is again we have to finalize the higher dose, still much higher dose. So again 4th time the dose will be given with a little increase in concentration. Then the escalation schedule for ascending dose needs to be considered carefully, using small increments of increase with higher risk drugs. If the drug is highly risk, like digitoxin, digoxin, they are very narrow therapeutic index drugs, right? So if the drug is given little point, a micron range also, if we increase the concentration, they are very, they produce very, very serious adverse events in the patient. So we need to be very careful in patients, high risk drugs like narrow therapeutic index drugs like digoxin and all. So we need to increase in a very, very less concentration, little slight increase only we can give and test in the patients. So we cannot give a suddenly increase from 10 nanograms to 20 nanograms and see the PK data and see the plasma concentration and how the patient is tolerable. No, we have to increase on a very small increments and then only test in the whether the toxicity is there or not, we are going to assess. And multiple ascending dose studies. Once SAD studies are proven that yes, the drug is very safe, and higher dose and safe dose, all these doses will be selected based on the SAD studies. Then we will go for MAD, that is multiple ascending dose studies. So here the main purpose of the study is to determine the safety at maximum exposures, but PK at these exposures is applicable to the design of next study in the drug development program. So here maximum exposures means multiple times we will be giving the drug to the patients or volunteers, and we will be testing how the drug is reaching the steady state and how it is showing effectiveness. Because most drugs need to be given repeatedly over time, like crocin. If the patient is having fever, we are going to simply one dose will not be sufficient, right? We have to administer every 6 hours so that the patient cannot will be relieved from the fever symptoms and all. So because most drugs are repeated, even diabetic, also diabetic patients, or in some other diseases, sometimes the drugs need to be given twice or thrice daily. So such time, in such scenario, the drug safety information is very, very critical. We have to see how the drug is reaching multiple doses, how the drug is showing its action and all. So the drug is administered for the number of doses required based on single dose PK to reach the steady levels and the highest exposure a given drug regimen will achieve, where the given drug exhibits first order elimination. So we are going to give continuously this drug, multiple ascending dose. So we will be giving multiple times and we are going to see how many times we have to give till it reaches the steady state level. Once the drug reaches the steady state level and shows this first order elimination, till that time we have to give the dose. So I'm going to show the graphical representation here. So single dose means only one dose we will see and test how it is effective and how it is safe. And multiple dose studies, we are going to give the dose multiple times, okay, so that the drug will be reaching the plateau, that is steady state levels. So this is how the study pharmacokinetics, single dose and MAD studies and SAD studies play an important role here in this drug development. So next one is food effect studies to determine if a difference occurs. So what is the interaction of the food? Why these food effect studies are to be monitored during the drug development program. So most of the drugs will be affected by the food, because once the food is given, the patient is taking along with the food, there is increase in gastric flow, and there are increase in the concentration of enzymes present in the GIT, and how they are going to show the effect of drug absorption, ADME pathway, how it is showing effect on the drug ADME pathway will be tested. So it is found to be, if found to be clinically significant, if the drug food is showing clinically significant interaction with the drug investigational new drug, that is food decreases the absorption enough to make it less effective, or that it increases absorption enough to cause toxicity. So if we find in all this PK data, once the PK data is given, we have to subject to statistical analysis also. And if it is proven that clinically significant interaction it is showing with the food, then either the food is decreasing the absorption and decreasing the effective dose to reach the particular concentration in the plasma to show its action, or it may be increasing the absorption also of some particular drugs and increase the toxicity to that particular patient. So that's why food effect studies ought to be performed. All these comes under early clinical studies, that is phase 1 trial studies only. All these studies will be performed. If the PK shows clinically significant food effect, adjustment of the therapeutic dose, then okay, it is showing some interaction. So what next step? What next step we have to take to prevent this food drug interaction? So adjustment of therapeutic dose or instructions on how the drug should be administered will be included in the approved drug labeling. So I think all would have seen many package inserts in the medicines, so whatever we buy. So what is this package insert says? They say all this PK data only. Whatever, whether the drug is absorbed, how much time it is taking, how the patient has to, how many times the patient has to take the medicine, in pregnancy can it be used or not, or if the patient is having any renal impairment or some special conditions, how the drug is showing effect. All these package inserts say, right? So from where all this data is coming from? Definitely during the trials and during the analysis of this pharmacokinetics, we are going to assess all this variability and with different population, and we are going to analysis statistically. And once it is all the data is ready, then we are going to instruct the patients how to use and how the drug will be in special conditions, how the dose should be taken. Everything will be included in the package insert. So here the same thing is shown here. If the PK shows clinically significant food effect, the adjustment of therapeutic dose or instructions on how the drug should be administered will be included in the drug labeling. Labeling only we need to inform that if you take the food, don't take along with the food, or take after 2 hours or before 2 hours, like that. All these instructions will be given in the package insert. So PK studies in special population, this is also very, very important in drug development. So because additional phase 1, all these comes under phase 1 only, what I was discussing earlier also and now also, like for first in human studies and food effect studies, PK studies in the special population, all this comes under phase 1 only, just to assess the safety of that particular investigational new drug. Okay. So additional phase 1 studies are performed to determine if PK differs in various special. What is the special population? Special population means there may be elderly patients, or they may be pregnant women, or they may be with geriatric patients or pediatric children, or like all this comes under special population category. A simple special population study can be used to bridge the entire drug development program of a drug for one population to apply to another population. An example would be a drug developed in Japanese populations that is then intended to be marketed in the US. So if Japanese people may be having different drug ADME pathways depending on the environment where they are living in and depending on the conditions, weather conditions where they are living in. The same will not be applicable for US. So because there also the patients, the amount of the food, cultural variations, food variations, and all these play a very vital role in variability in PK data. So that's why we need to see in Japanese population, then intend to be marketed in the US. So all these special population category studies also to be conducted before we go for phase 2 and phase 3. So most small molecule drugs are investigated in subjects with hepatic impairment, that is liver failure or kidney failure patients, because we may be knowing kidney failure patients, there are a number of proteins will be some albumin contents and creatinine levels all will be more, which will show the effect of in turn affect the drug ADME pathway. So here hepatic or renal impairment, depending on the drugs intended, elderly patients, obese patients, certain racial or ethnic groups, or others are performed. So we had to perform in other special population also under phase 1 before we go for phase 2 and phase 3, so that how the drug is showing variation as per the ethnic culture, races, obese patients, all these PK also we are going to estimate in the drug development program. In hepatic and renal impairment, plasma proteins such as albumin can be lower than in healthy subjects. So free drug concentrations are often examined to determine if any PK differences are there to the differences in binding. If the plasma protein variation is there, definitely the drug protein binding capacity will change. So when the drug protein binding capacity will change, accordingly the drug distribution, the amount of drug in the plasma or in the urine, everything will be showing variations. So sometimes the drug gets increased in concentration, or sometimes the drug will be decreased in plasma concentration, which results in significant drug difference. So PK studies in special population also to be studied before we go for phase 2 and phase 3. So drug-drug interaction studies, which is the very, very important one apart from this food, along with the food drug interactions, we have to assess what is the concomitant drug interactions. How, I mean, if we give simultaneously the investigational new drug with the other drugs, how it is showing variation or how the PK is altering. So PK data, a small number of subjects exposures to one or both drugs can determine if there is a safety risk by examining previous PK measurements and correlated safety findings. So we know in the absence of any other drugs, how the drug is showing PK data, we will be getting in phase 1 simple studies. But in the presence of other drugs, how it is showing variation, that study designs we have to see. If the drug is showing any potential interaction, and the drug, usually the drug under development is dosed to steady state and at a therapeutic dose, and we had to see how the drug is showing interaction with the other drugs. Generally, these drug-drug interaction studies will be conducted on a small number of subjects, healthy subjects, around 12 to 24, and confined to a research clinic, and consists of a fixed treatment sequence for all the subjects. And here, if you see, an example of common design would be where the probe drug means when the patient is taking other drug. Probe drug means other concomitant drug, which he is going to administer for different purpose. Probe drug is administered alone, followed by a washout period. We need to give for a certain time for washout. Then the drug under study, we have to give our investigational new drug. And until the steady levels are reached, then the probe drug is co-administered. So before we had to give one probe drug, then we need to give the washout period, then we give our IND, then till it reaches steady state levels, and again probe drug is co-administered along with that. So PK of the probe drug is measured to determine if the PK is affected. Probe drug pharmacokinetic data also is equally important, because we are going to assess how it is showing how it is changing the PK data of the IND, investigational drug. The sequence could be reversed if an investigational drug is hypothesized to be affected by the probe drug. If we feel that there is an interaction, we have to change the sequence also. First, we give the IND, then give the washout period, then probe drug is given, then estimate whether any interaction is there. Hope all of you are active and listening, and because no doubts and nothing is visible in the chat section, I feel all are listening very actively, right?
Yes,
so, and apart from this PK studies, where and all these pharmacokinetics applications we use. phase, we discussed how the drug development program, how it is showing everything. We discussed like early clinical studies, first in human studies, food drug interaction, drug interaction. All this comes under phase 1 trials, and small number of patients we test so that the patient's safety will be analyzed. Once the drug is proven very safe at the given dose, then we will go for phase 2 and phase 3. Phase 2 and phase 3 also, once the drug is successful, then it will be released into the market, right? So here, where and all this other pharmacokinetics data is applicable, and where, how we are going to evaluate. So here, if you see pilot studies, PK studies afford the earliest look at data in humans. So PK studies, if we have all the data, then we can just see how it is showing effect in the humans. The pilot PK study serves as early look at the early look and has the potential of saving time and money as well as informing pivotal studies. If the sponsor, means who is going to sponsor for this entire drug development program, chooses a pilot study in a small number of subjects can be carried out before proceeding with a full BE study, bioequivalence study. So before going into completely bioequivalence study, why we are going to perform that, we will discuss in the next slides. Okay. So how and from where should I start my research? The question will be, conduct a pilot study so that you will get all the answers. So if we are going to assess in the pilot study, if the drug is showing fully, completely active based on the PK data, so application of pharmacokinetics comes under pilot studies also. Okay, so we are going to test the in a small number of subjects how this is effective. And in applicational new drug applications. So here, if you see, PK studies are best known as those relied upon to prove the bioequivalence to the reference listed drugs. Bioequivalence means the drug will be the generic drug will be whatever the drug is, like paracetamol is showing same crocin under the brand name crocin is equally pharmaceutically equivalent, and as well as it is therapeutically equivalent to the Dolo 650 or whatever the other brand name. Means reference listed drug and our investigational new drug, both are similar. That is nothing in not only in action, either pharmaceutically also, and it shows same AUC, same Cmax, Tmax, everything matching, means then the both drugs are called under called as bioequivalent drugs. So here, if you see, PK studies are perhaps best known as those relied upon to prove bioequivalence to the reference listed drugs for generic new drug applications. ANDA approvals require evidence that a generic drug product has the same active ingredient, dosage form, strength, route of administration, and conditions of use, that the proposed product is bioequivalent to the reference listed drug. So we had to submit, during submission only, we had to show that the drug is equally effective, pharmaceutically equal, and therapeutically also it is equal. And we have to give all the PK data here so that they will see, okay, if the drug is same pharmaceutically also and same active ingredient they are using and same dosage form, same strength, everything they are using, means PK data also will be matching, right? So, and the drugs are bioequivalent, then they give approval easily. So for NDA applications also, this PK data is very, very important. Next, bio-waivers. Waivers means they give waive off means they give discount, like completely no need to conduct the further studies on this particular. For what is this bio-waivers, where it is applicable, why PK pharmacokinetics is useful in this bio-waivers. So here, anyone submitting an NDA, including NDA, may request the FDA waive the requirement of evidence measuring in vivo bioavailability or demonstrating in vivo bioequivalence of the drug product that is the subject of application. So we are going to perform on the same generic drug. So please give us excuse, so that no need to perform the bioavailability studies and bioequivalence studies that they are going to apply to the ANDA. So underlying circumstances of waiver requests are many and varied. For example, in vivo bioavailability of bioequivalence product may be self-evident. So if the product is equal, pharmaceutically same active ingredient, yes, definitely they are bioequivalent, right? So this is generally applicable to injectables, ophthalmics, otic solutions, inhaling gases, topical solutions, nasal solutions that contain an active in the same concentration and dosage form as a drug product that is the subject of approved NDA. If the drug is already approved and one person is manufacturing with the same active ingredient, same dosage, same strength, everything is same, then no need to do the all the PK data again. They can waive off the requirements, whatever they are asking for. So formulation changes that significantly affect the absorption or local availability will generally rule out a bioequivalent. So formulation changes which affect the absorption or local availability, like local application, like topical formulations and all, they rule out a bioequivalent. Biosimilars. Here also PK data is very important. Though we know proteins, what are these biosimilars? They are applicable for protein molecules. Protein molecules are manufactured from living organisms, and they are very, very large and complex molecules compared to conventional chemical drugs. Not like unlike a normal chemical mighty. They are very, very large and complex protein structures. So for them, how we can say that they are similar? If some person is manufacturing insulin, with the same some other source from some animal or something they're extracting, they cannot say that this drug is similar to the particular protein already existing in the market. So biologics tend to be made up of proteins, carbohydrates, nucleic acids, or a combination thereof, and also include the cells or tissues used in transplantation. Because of this relatively complex structures and method of manufacturing, biosimilars are never identical to the brand name product. So, but it will instead be shown to be highly similar. We cannot say that this product is same that of the generic product. So we can say that yes, there is a highest similarity is there, but not biosimilar. So for such products, what we had to do is analytical studies demonstrating biological product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components. For example, if you see animal studies and a clinical study, that is immunogenicity and pharmacokinetic and pharmacodynamic sufficient to demonstrate the safety, purity, and potency for the conditions of use in which the reference product is licensed. So if we give all the PK/PD data and we can give all the immunogenetic studies and all to the regulatory authorities to the reference product, then they may consider this as a highly similar, that is biosimilar. They may just consider for the further approvals. So these pharmacokinetic studies play an important role in bridging. What is this bridging? So PK studies are useful in the development of bridging the data. So here, if you see, bridging is narrowly defined by ICH guidance as study performed in India. We are testing some simple example, and we are testing a paracetamol effect, and all the PK data we collected, it is very effective, very safe, very efficacious. Okay, the drug is released into the market, and we are going to assess in post-marketing also, everything is done. But how this paracetamol is showing effect in a new region like Europe or US? So if we had to test that, our PK data will be giving the information, yes, this is proven. Seeing our PK data, they may give approval to that particular drug in that particular country. So a study performed in the new region to provide pharmacodynamic or clinical data on efficacy, safety, dosage, and dose regimen in the new region that will allow extrapolation of the foreign clinical data to the population in the new region. So from one country to other country, also this PK data is very, very important for filing for the for approvals, regulatory approvals. So we had to submit all the PK data. Then only they will be seeing the whether the drug, they can give approval in the particular country or not. They're seeing our PK data also. For example, here some examples I have given here. Sandoz developed its own version HX575, which was approved as a biosimilar in Europe in 2007. The trade name of HX575 is Vinocrit, which relied upon Eprex, which is the reference product. Reference product is European formulation. So that is Eprex. Sandoz went on to compare to US, European, and HX575 formulations as follows. 3 formulations they have developed, that is US one, European, and HX575 under 3 different brand names. The study was conducted in 2 parts. Part A compared European marketed HX575 and US marketed Epogen Part B compared European marketed Aperex and HX575 manufactured at two different drug substance production sites. Production sites are also varying. So one is manufactured in Europe, another is manufactured in US. So bioequivalence was shown in all pair-based comparisons. So the products are identical or similar in all the PK data. So that is bioequivalence also, it is shown. Thus the bioequivalence and pharmacokinetic equivalence allows bridging of the data from the clinical studies using either US or European apoetin alpha products. So they compared the data which is collected in the US and Europe, and they did the bridging of the clinical data. Because of the similarity of all the three products, despite the different formulations, clinical data produced with a product sourced in either region could be used to support further marketing authorizations, including in other geographical markets. So because here in India, it is a tropical country, it shows different variations in the climate. The same US, if you see, there is a different change in the climate, and the same there is culture variation also. Everything is the variation. So if we give the PK data, and it is going to help and bridge the gap between the two studies, and they are going to give the regulatory authorizations to whether the drug will be allowed to release into the market in US or Europe also will be tested if the pharmacokinetic data is given to the regulatory authorities. So I hope I could cover all the applications of pharmacokinetics, and the last part I came here, the summary. So in starting from the brief to the preclinical, how this pharmacokinetics, where it is applicable means it is applicable in starting from the preclinical stage till the end of the, till the ongoing process, that is under phase 4 also, pharmacokinetics play a very, very vital role. And if you see the PK measurements in the first in human study provide confirmation of safety at the measured exposures from the tested doses. So even if exposures from a given dose change due to food effects, drug-drug interactions, drug-disease interactions, or use in a special population, safety can be associated and risks assessed will be by bridging these results to the initial safety or efficacy exposures. So if we find any food effects or drug-drug interactions or drug-disease interactions, or how the drug is showing PK variation in the special population with hepatically impaired patients, renally impaired patients, in pregnant women or in children, how it is showing variation, all this safety can be associated. And if any risk is there, we can assess by bridging by reducing the dose or by just giving instructions to the package insert and all that, by bridging these results to the initial safety or efficacy exposures. So throughout the drug development program, pharmacokinetics play a very, very vital role to link exposure to efficacy and safety. And also it assists in the determination of dosages of marketed drugs. For this reason, PK data are important for a part of the information provided to the clinicians. So without PK data, I am telling the drug development cannot happen. Simultaneously, without bioanalysis, pharmacokinetics interlinkage, and without any department not functioning, we cannot give any data, and the drug development program cannot just happen like that with only one department. So pharmacokinetics, bioanalytical department, R&D, all this will play a very vital role during the drug development. And pharmacokinetics, as I told you, from the preclinical studies till the drug releases into the market, phase 4, and post-marketing surveillance, once the drug is released into the market also, we have to continuously assess the safety and efficacy of the drug, and it will be monitored throughout the marketed formulations also. So this is how I conclude today's presentation, and these are the references. And if any doubts, please question me. I'm ready to clarify all your doubts, and I'll be very much happy to clarify your doubts. I request all the participants to share your views and to just discuss if any questions you have.
Thank you, ma'am. Thank you for your wonderful information and educating us. And I'm here with a few questions for you, ma'am.
Sure.
does the pharmacokinetics, the parameters, all these drugs, it increases the sensitivity in infants?
Infants?
Oh,
Yeah, ma'am. That will be actually that will be tested in a special population in phase 1 only, phase 1 trials. So they will be given very, very low dose to the patients, infants, and they will be seeing in special population. As I told you, all this infant study also comes under special population. So they will be testing at a maximum safest dose will be given to the infants, and then they will analyze whatever accordingly. And if the infant is not tolerable, then they have to decrease the dose. Sometimes there is a chance of death also, or sometimes rare events will happen during these trials. But that's how once if the drug has to come out into the market successfully, some sacrifices and some things will be considered here, though it may happen, but they will try to give the safest dose possible to the, especially to the infants, because it comes under special population study. So it will not be like a regular population study, the special population. So they give the very, very safe dose only, whether based on the preclinical studies, they get the information.
okay.
Hope I clarified.
Yes, ma'am. And will the volume of distribution affects the area under curve of any drug?
No, not directly. Area under curve is nothing but the bioavailability of the drug, means how much of the drug is completely getting absorbed. How much drug is getting absorbed comes under this AUC. So if you see the volume of distribution, how the drug is getting distributed, that is the second during distribution phase, it will go. So AUC is completely not directly linked to the volume of distribution, but definitely it will affect in the form of like if we go for the calculation, there is a small change will be observed with the volume of distribution also. If the drug is distributing very fastly, then the drug absorption, once it absorbs, immediately it will start this elimination phase. So that time we had to see how this is showing effect on the bioavailability, how the volume of if the drug is binding very plasma protein binding is happening almost to 95 to 96%. Definitely how this is impact you see, we had to assess during pharmacokinetic evaluation.
Okay. Okay. And the other question is, what is the purpose of single ascending dose study in preclinical?
Yeah, preclinical, it's not preclinical, it is early clinical studies. Preclinical means we do testing in animals only.
Yes.
So their single dose study and all, we are, I did not focus much, because all these animal studies we can do on cell lines also, and all these biological fluids also. Here, especially I focused on this trials study, phase 1, under phase 1, early clinical studies. Single ascending dose SAD studies are to be performed just to assess the safety of that particular drug. So we will give single dose only multiple times. Once the drug is, we will give the best dose selected based on the preclinical study. What is the best dose to be given to the humans? We will decide. So based on that data, we will give that much dose to the patient. If the patient is tolerable and it is not showing any toxic reactions to the patient, then we will give the dose multiple times to assess how it is till it reaches the steady state levels. That is called single ascending dose studies, just to assess the safety and whether how much dose it is showing toxicity. For that purpose only, we will be testing this SAD study.
Okay. Okay. And another question.
Yeah.
You have discussed about 505B section B2, right?
Yes. Yes.
So what is the difference between ANDAS and that 505B2?
This is application of new drug application, that is which is under what I can say is when we are applying for bioequivalent drug product. Like we are going for already generic product is there in the market, like what I told you, like paracetamol, and the same product will be manufactured under a different brand name. So, and we are showing this is bioequivalent and the same, we are manufacturing with the same content, same active content, then definitely this section will be under this section, we had to file for regulatory.
Okay. Okay. So how many years of exclusivity can be granted under this section 505(b)(2)?
Exclusivity depends. It depends on how the PK data can satisfy the regulatory, and it may vary. I'm not sure particularly how much exclusivity, maybe like 10 years, 5 to 10 years they give. I'm not sure exactly, because this comes under regulatory department. Maybe I think it is like around 10 years they give. If everything is bioequivalent and it is the same pharmaceutical equivalent, and therapeutically also it is bioequivalent, if it is proven, and if the regulatory data is submitted to the authority, then only they will give this exclusivity.
Okay. And here is another question in the chat box. What are the possible side effects or risks of the new treatments of clinical trials? They have posted this question.
We cannot say just that possible side effects or risks. Definitely there will be so many side effects or risks to the patients. That's why they should be completely, all these trials studies should be performed under good clinical practice. The GCP trained practitioners only can be given this chance of testing the IND drugs to the patients, and they had to sometimes some patients may die also. It may lead to death also. All the rare adverse events, serious adverse events, everything should be reported. Definitely there will be a risk, and it should be, and only one thing, one advantage of this phase trials is we will be informing to the patients, and we will be taking the consent from the patients also that this drug we are testing, yes, this is safe also, but sometimes it may show any interaction in you. So it may happen, this death also may happen. That everything we will inform the patient, and we will take the consent from the patient. That is informed consent form, we say. If the patient, like children are there, their parents, guardians, whoever are there, we need to inform everything regarding IND, and we have to take the consent from that particular patient. If the patient is completely satisfied, yes, let me participate in the trial, so that whatever happens, let it be, but it will be useful for the society. So definitely those participants only will be doing actively participating in the trials. And there are side effects. I cannot say there are side effects, and I cannot say there are risks. There are sometimes rare adverse events, sudden death also will happen. And I hope you would have known for COVID study. Even I was the volunteer for COVID study, and I was during this Covaxin only, I was a volunteer. So though we know the side effects or interactions, we want to do something to the society. So after consent, then they will give the so I tested before the vaccine came into the market. Only me and my husband both were volunteers, and we took the Covaxin. So like that, if any patients will be completely informed, there will be risks, there will be side effects, but some sacrifices are there. In once the drug should be successful in releasing into the market, means so much hard work and so many risks should be assessed during the trial study.
Thank you, ma'am. Thank you for accepting our invitation, and thank you for addressing us.
Thank you. And any doubts from any students?
Yes, ma'am. All these questions I have got from the students itself.
Sure. Okay.
They are
glad to clarify their doubts.
Yes.
Thank you, ma'am. Thank you for your valuable session.
Thank you.
On behalf of KVSS Corps, I would thank every participant for this session, for listening patiently. Thank you, ma'am.
Thank you so much for giving this opportunity to Siddhartha College of Pharmacy and Dr. Vijaya for inviting me to present this lecture. Thank you, everyone. Thank you, all the participants.
Okay. You can leave the meeting.
Can I leave the meeting?
Yes.
Yeah.
And I request all the participants to fill the attendance form